just in English, I'm really glad that you're here in Germany, here in Trier, and thanks a lot for this.
Thank you, Sebastian, and my true apologies. I don't speak that much German. We had it a little bit in school. I come from Belgium, so I understand it. I'm Flemish, so it's very close to German. Yeah? Okay. So it's very close to German. I understand a little bit when you speak, but it's very difficult for me to speak German. So I apologize. I'm going to do it in English, but it will be translated and subtitled. So I'm very happy to be here. Thank you, Sebastian, for inviting me. It's also amazing to see so many families and especially so many small children. So I can imagine that much of my talk, it will be maybe the first talk about KCNQ2 mutations, and it will be a lot of information. I'll try to explain it in as easy way as possible, but if there's any questions, you can surely come to me afterwards and we can discuss, and I will try to answer all questions then. So what I'm going to try to do today is a little bit to talk about the whole spectrum of KCNQ2 related disorders, because you already see it here. There's many different children with many different problems. You probably know that sometimes mutations can also give a very benign disorder that we call benign familial neonatal epilepsy. So it's a big spectrum, and the difficult thing is when you get this diagnosis, you get this question: what can I expect? What will come, and how will my child develop? And knowing what your variant or the variant of your child does, it can help already a little bit. It does not tell you everything, but in many cases it can at least make the distinction between this benign familial neonatal epilepsy and the more severe form. And as we'll see, there's also two different types of mutations: loss of function mutations and gain of function mutations. And we will discuss this a little bit. So first of all, I put here a slide to say, because we know, and it's also here, I think, it's a rare disorder, KCNQ2, DEE of epileptic encephalopathy. But in all the genetic epilepsies, it's one of the most frequently mutated genes. So for example, what you see here, you don't have to look to all the details, but this is from a study that looked at all the papers and studies that have been published that look at the results of genetic testing in patients with epilepsy. And maybe it's very small, I don't have a pointer, but the first most frequently mutated gene is SCN1A. You might have heard of patients with Dravet syndrome, that's the gene. And the second one is KCNQ2. So the second most frequently mutated gene in genetic epilepsies is KCNQ2. But again, this is all types of KCNQ2 related epilepsies. There is a study in, I think, Scotland that looked at all children below the age, I don't remember the age, I think 2 years of age, that were born, and then they did genetic testing, and they saw how many of the children that they were born in Scotland with epilepsy have, for example, KCNQ2 mutation, and they found that 1 in 70,000 children that were born in Scotland had a mutation in KCNQ2. As I said, there's the benign one and then the more severe ones, and the more severe ones, they're called, and we discuss this later, KCNQ2 developmental and epileptic encephalopathy. They estimate that around 1 to 3 per 100,000 children have this disorder. If you only look at neonates, so these are very small children with epilepsy and the first weeks of life, then 1 in 3 children with genetic neonatal epilepsy have a mutation in KCNQ2. In KCNQ2, yes. So this is actually a lot. Obviously, not all children with epilepsy have a genetic form, but of the genetic neonatal epilepsies, 1 in 3 has a mutation in KCNQ2. KCNQ2-related epilepsy also has a long and a short history. Why do I say long? It was one of the first epilepsy genes that was ever described. in 98. So in 94, it was the first gene that has been described, and then in 98, I think KCNQ2 was one of the second or third, epilepsy genes that were described at that time in this benign familial neonatal epilepsy. But then it took until 2012 when we discovered that sometimes mutations are not that benign. There's a more severe form, KCNQ2, developmental and epileptic encephalopathy. In 2012, we said it's always children with neonatal seizures. And in 2017, we first saw that some of these children with KCNQ2 mutations do not have neonatal seizures. They do have neurodevelopmental problems, but not always seizures in the first weeks of life. So you see, we know KCNQ2 for a long time, but this severe disorder, it's known for 10 years now, and we're still learning a lot. And there's still a lot that we don't know. At first, of course, it was very strange for us to say, how come some children do totally well and others don't do so well, and some have neonatal seizures and some do not have neonatal seizures? The way we are understanding why this is so is because collaborators, other people, are able to test what the mutation does on the function of the KCNQ2 channel. They do this because obviously you cannot test this in the brain of children, that's too difficult. So what they do is they take the protein, KCNQ2, they make it, the protein, they put it in another cell type. For example, they often use oocytes of frogs, and then they test how the current through the channel works. Is it more potassium current or less potassium current? And now we know that these different types of KCNQ2-related epilepsy, they're caused by different mutations that have a different effect on the channel function. And of course, then the question comes, do all these different types of mutations and different types of epilepsies and children, do they need different types of medicine and different type of therapies? And this is something that we're trying to understand, and more insights are being generated in the last few years. Before talking about the whole KCNQ2 and the different mutations, I shortly want to say something about what is a gene and what is KCNQ2, what does it do? Just as a reminder, so KCNQ2 mutation is a change in your DNA. And your DNA, it's like a genetic code. It's a code, it's like a handbook that every single cell in your body has, and it has a code to make different types of proteins. And it has a code to say to your cell in your body how it has to function. It has a code to say you're a brain cell and you're a liver cell and you have to do this, and so it's really essential. It's like a long strand of all different molecules. There's 4 different molecules, A, T, C, G. We call them nucleotides. And these are the letters that you see on your genetic report. And any of these letters, they can change, and that is a mutation. And DNA in each of our cells has 3 billion of these nucleotides, or these letters, A, T, C, A, T, 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 A, A. And this is a code, and sometimes changes occur and mutations. Now, what is a gene then? Gene is a part of your DNA that has the code for one single protein, one single protein in your body. Three of these nucleotides together, they have the code for one amino acid, and several amino acids next to each other make one protein, and this protein does something in your body. We all have 2 copies of each gene, or most of our genes. One comes from the mother and one comes from the father. And in some genes, if you have 1 copy and the other copy doesn't work, that's not a problem. For other genes, as soon as you have a problem in 1 copy of the gene, then you get a problem and you get a disorder. So there's different types of genetic diseases. In KCNQ2-related disorder, one copy that doesn't work already gives you problems. So there's always a healthy copy from the mom of the dad, but still you have a disorder. If you ask, like, why do some mutations occur? Well, I told you, like, every single cell in our body has the same DNA, this strength of 3 billion of nucleotides. Then you have to realize when a child is made, an embryo is made, there's an egg cell and there's a sperm cell that come together, and then this one cell, it has to divide millions and millions and millions in time until it has a whole body and a whole human being. For every cell, this DNA is copied, but now and then an error is made in copying. It's like writing a book and by hand, and sometimes a mistake can happen. And our body has a lot of systems to repair these mutations or these changes, but sometimes they don't manage to correct it, and then you have a mutation. And that's how sometimes de novo mutations also occur. You don't have the parents are totally healthy, but when the little baby is made and the body is made, an error occurs. That's a de novo mutation. And that's, I think, my next slide. You have genetic disorders where a parent carries a problem, a mutation. For example, in the first picture, there's 2 copies of a gene and a chromosome, and the father or the mother can carry a genetic disorder, and then he passes it on to his children. That's an inherited mutation. Or the mutation can only occur when the baby is formed, or this is a de novo mutation. And as we'll see later, most of the variants that give this severe KCNQ2 related disorders are due to de novo mutations. In theory, when this child of the second figure will have children, then it will become an inherited mutation because he or she has a chance to pass it on to their children. But many of the children with KCNQ2, the severe form, epileptic encephalopathy, will never have children, so it remains an isolated mutation. So then we know what the gene is. So what is KCNQ2? KCNQ2 is a gene that has the code for a potassium channel. A channel in the brain. I don't know if maybe my mouse, you see it? No, I don't have a pointer. So maybe I do walk a little bit there. So you have this, at the bottom you have a nerve cell, and at the border of the nerve there's a membrane, and in these membranes are channels, all different types of channels, and one of the channels in the nerves is KCNQ2, or the subunit of this channel is KCNQ2. So it's a channel, and when it opens, potassium goes out of the nerve cell. And why this is important? Because of the way that nerve cells in the brain communicate with each other. One cell communicates with the other because there's an electric signal that goes all the way over the cell, and at the end of the nerve cell, there's neurotransmitters, that's chemical substances that are exposed. They connect with the next cell, and then another electrical signal can occur. So through these electrical signals, nerve cells communicate with each other, and to make this electrical current, or more in particularly to dampen these electrical currents, the potassium channels in your brain are very important. It's a very complicated mechanism, but in general, sodium channels, the one that you see behind it, they lead to the occurrence of electrical signals, and potassium channels, they make sure there's not too much of these electrical currents. So it's like a break in the nerve cell. They make sure that your nerve cells don't become overactive. So it's very important. You can imagine when this break doesn't work, then the cells become hyperactive and you get, for example, epilepsy. So this is a little bit the structure, all at the left. All these blue dots, those are amino acids, and all together they make a protein. The figure with the green and orange, it's a little bit more easy representation. So it's a protein that goes through the membrane. It has 6 of these transmembrane domains. One of them, the 4th one, you see these pluses, that's a charge. So these amino acids, they have an electrical charge, and that is the voltage sensor. So it senses how electrically charged the neuron is. And the orange one, it's the POR loop. So they together, 4 of these subunits, and that's the second figure B, they come together and they form a channel. And through the orange part, ions, in this case potassium, can go out of the cell. And you see below the channel that can move in the membrane and it can open or close. And when it opens, potassium goes out of the cell. KCNQ2 is one of the subunits of these 4. And sometimes it makes a channel with other KCNQ2 subunits. It also can make a channel together with KCNQ3 or KCNQ5. These are other related subunits of the channel. It's important to realize KCNQ2 is in every cell of nerve cell in your brain, and it's already there from very early in life, in fetal life, so when it's still in the womb, in the embryo. This already shows you that it's a very important gene. That is probably already important in neurodevelopment and in making nerve cells connect to each other. And that's probably, but we don't fully understand why yet, if it doesn't work from very early in life, that the nerve cells or the brain doesn't develop well and you get neurodevelopmental problems. So then go back into the history of KCNQ2. I said in 1998, it was described that mutations, certain errors in the gene, can lead to epilepsy. And these were the kind of families that were described then, like the mother and then the grandmother and then the son and then his child. They all had neonatal seizures, so seizures when they were just born. But they had it for a few weeks and then they stopped and the child was totally normal, the EEG was normal, the MRI was normal, so it was a very benign disease. It was frightening for parents because they saw their child seizing, and you all know what this means. But then the mother can tell them, I've seen it, you had it too, and it went away. So it's something benign. You see the EEG there, that's the EEG of a very small child, a neonate. This is a normal EEG, so also the EEG in all exams were normal. This was in 1998. A lot of mutations have been described for BFNE, benign familial neonatal seizures, and many of them have been tested already in a cell system. So it's really in a dish. They see how we make this mutation in the protein, we put the protein in a cell, and we measure the current that goes through the channel. And they saw that the current, so the potassium that goes through this channel, that there's less current. And the amount was about 25 to 50% less of current goes through potassium. And we saw before, potassium, this current, it's like a brake. It reduces the excitability of the cell. So if you have less of this current, you have more hyperexcitable nerve cells, so you get epilepsy. So this was clear, and we thought we understand KCNQ2, and this is it. If there's a problem, you get epilepsy, but that's it. But then in 2012, we saw that some mutations in the gene, they did not only lead to epilepsy, neonatal epilepsy, but many of these kids developed problems afterwards. They didn't sit up well, they were hypotonic, some didn't walk, didn't speak. So this was not benign familial neonatal epilepsy, obviously. And this is a little bit what most of these children have in common. These children, they had neonatal seizures, so in the first days or weeks of life. Also, just like in BFNE, they have often seizures where they stiffen up, and you might probably recognize this from your children. Sometimes they do a little bit of jerking afterwards. Many of them turn blue when they get this. They might stop breathing. The heart might be beating slower, and then it stops. And often it's like multiple times a day in the beginning. If you then do an EEG in these children, it's not normal. You see there's like a burst, and then there's nothing. And there's a burst of activity, and there's nothing. That's what we call burst suppression on EEG. Not all of the kids have it, but many of them. And then later on, even if in some of them the seizures go away with the right treatment, but they have neurodevelopmental problems. They don't develop like other kids should. Often there's also a range of other comorbidities. Some people have difficulties swallowing, they have obstipation or constipation, bowel problems. They seem to have some discomfort. If they grow up, some have some behavioral problems, a little bit autistic, not all of them, because some are very happy. But it's not only epilepsy, that's clear. So then obviously this was the very beginning. We saw these children, but we didn't understand yet why do some variants lead to a much more severe epilepsy. If we look at all the differences and we compare both types of variants, we know that in the first, the benign ones, it runs often in the families. So it's often an inherited mutation, although not always, because and every mutation has to start somewhere, right? There's all types of mutation all over the protein, as you can see here, there, from the early beginning to the end. Sometimes the whole gene or one copy of the gene can be just missing, while in the more severe end of the spectrum, it's all missense variants, and this means there's only one nucleotide that is changed, and they cluster in specific, very important places in the protein that are very important for function. There's the charged membrane, the fourth one, or the pore, are at very specific parts at the end of the protein. At the very beginning, very early, I think one of the first American Patient Organization meetings, I met up with Ed Cooper, that some of you might know. He works in Baylor. He had been working on KCNQ2 for years before it was associated even with the disorder. And there was another collaborator in Italy, Maurizio Tagliatelli. He also did a lot of this variant testing in these oocytes. And we thought we have to put all these variants together, and maybe if we see patterns, like that specific variants are associated with a specific phenotype, and then we can select them for testing, we can find why there's such differences. So in the meantime, using the information that really comes from families, right? They said, my child has this variant, and he doesn't develop normal. Many of these variants have been tested, and what they saw, not I, but my collaborators, in this graph below, you see the current, the normal current through the normal channel, and then the current through the mutated channel. And you see that it's more than 50% reduced. So there's really very little current that still goes through the channel. And this is what we call dominant negative variants. So what does this mean? Remember that there's four subunits that form the channel. Apparently, these mutations that lead to the more severe variant, they just not only not work well, but they also act in such a way that the good copies from the other parent also cannot function that well anymore. So you have really less than 50% of the current. So it's a more severe loss of function than the benign ones. And this is really related to which variant you have. So in general, it's really the variant that your child has that defines whether he has a good outcome or a more severe outcome. Obviously, there's always exceptions, and we all are different. There's other factors that will influence how your child will develop, but in general, it's really the mutation. It is the cause of most of the problems. Let's put it that way. Okay, I said this. In the meantime, so this was, I don't know, maybe 2014 that we started to understand this difference. In the meantime, a lot has been described, again, with help of parents, like to which drugs do these children respond better. And I think what is already very clear for treatment for seizures in general, most of the children, I say most because not every child, respond better to anti-epileptic drugs that we call sodium channel blockers. So they act on the other channel that's very close to KCNQ2. This is, for example, a study that has been done in these big families with BF&E. And they looked at how fast the children were dismissed from the hospital. And of course, the sooner that the seizures stop, the earlier the parents can go home, right? And they saw if they started carbamazepine, this is a sodium channel blocker. I think in Belgium it's called Tegretol, maybe in Germany too, I don't know. If they started within 3 days, the child could go home like after 3 days almost. If they did not give carbamazepine or they started later, the child had to stay in the hospital much longer. So this already showed that if you give a sodium channel blocker or carbamazepine, seizures go away sooner. So this was in these big families with the benign form. And in the meantime, I just put the titles of the manuscripts. We know that also children with severe KCNQ2 developmental and epileptic encephalopathy often respond better to sodium channel blockers such as carbamazepine, oxcarbazepine, trileptal in Belgium, lamotrigine, lamictal, difantoine, phenytoin. So they seem to respond better, seizures. We don't know totally why it is, but probably remember that KCNQ2 is the brake. If the brake doesn't work well, the nerve cell starts like being active, and this activity it's these sodium channels. So these sodium channels, they open more, and sodium channel blockers, they block the channel that's next to it. So they really work together, and probably that's why they work better for seizures. Again, not in everybody, but in many of the children. I already can say now that although we have the impression that if you treat the seizures well, the children will do better, but you don't solve all the problems. Seizures are not the only reason that the children do not develop normally. There's something else that the channel does, why these children does not do well, why these children do not develop totally normal. So you don't solve every problem, but at least it helps to control the seizures better. You might probably also have heard of retigabine or ezogabine. This was a drug, Trobalt in Belgium, Potica in the States. I don't know what it was called in Germany. It was on the market until 2017 as an anti-seizure drug. And it acts specifically on these potassium channels with the KCNQ2 subunits. And it's make sure that they open more. And this was logic, because it's the brake. So for epilepsy, it's too much excitability, so you make the brake work better. So it worked for epilepsy in other patients. It was not given specifically for genetic epilepsy, just for general epilepsy patients. And at the time, it moves by itself. Okay. At that time, 2014, it was still on the market. One of our colleagues, again, he tested in a dish the current through these mutated channels. The white spots is the M current when the channel is mutated. And when they added retigabine, you see that there's much more current going through the channel. So it seems to help for the channel function. And indeed, at that time, several of the KCNQ2 encephalopathy children, they have been treated with retigabine or ezogabine, not in a trial, just because doctors thought it might be useful. And we saw that indeed, many of the children, the seizures responded. They had less seizures. Some of the parents indeed also said, my child is more alert, although this was not really a clinical trial, so they did not do neuropsychological testing or whatsoever. But then in 2017, the company took the drug off the market. And this had several reasons, among which also economical reasons, because the drug was not used that much. Also because it had some side effects, like it often gives urinary problems, but because it also acts on a channel that's on the bladder. And in long-term use, they saw that sometimes there was some blue discoloration of the nails and the retina in the eye. So they took it off the market. Now there is a trial, and we'll talk about that later, the EPIC trial that's currently ongoing, because the company Xenon, they made another formulation of it, so little children can take it, but because before it was only in the tablets. Now there's like a granulates, like granules, I don't know how you say that in English. So they make it something that it makes it easier for small children to swallow. And they try it specifically in children with KCNQ2 encephalopathy, but we'll talk about that in the next talk. Since then, again, a lot of people are working on this disorder, and I think one major advancement also that we have made in the meantime is that a French colleagues, I think 2 years ago, they made a mouse which specifically also had a variant of KCNQ2 encephalopathy. Also, these mice, they have seizures. They also have cognitive problems. They can test it, they can have them do all kinds of tests. So now they have this mouse model is available also for other groups in the world to test, for example, a new drug that they develop to see how does it work in a mouse, does the mouse tolerate it well? And this is a necessary step before you can try it in children. You have to find evidence that your new drugs work. And it's also very useful to understand exactly what happens in the brain, because we cannot just take a piece of the brain of the child and look what happens, but this is something that you can do in a mouse, for example. So there's still a lot answers that need to be answered, but we thought we really understood a little bit what made children being so different because of the mutation effect. But then I think, let me see the date, 2015, my colleague Maurizio Tagliatella, so again from Italy, he measured again in a dish, he measured other mutations that had been described in literature. And you see, yeah, there's always these letters, right? And position, the number in the middle, it's the position in the protein. So there's a whole strand of amino acids. So the 201st amino acid, was an error. So he tested these variants, and what he saw is that when you have these changes, the channel works too much. They're almost constantly open. So there's constantly potassium going out of the cell. So apparently this is also something that can happen. And what we again then did through the registry, we contacted physicians and parents with children with these variants, starting with the variants R201C, R201H. And we looked at what the phenotype, what did these children do? And we saw that all of them, they also had strange movements when they were just born, but not this tonic stiffening. They were more like jittery, constantly moving like this. Often they thought it was epilepsy, but it didn't seem to be epilepsy when you look at the EEG. And then later on, they developed spasms, so West syndrome, something that some of you might have heard of, and they had a very profound delay. They all had the same pattern, but they didn't have these neonatal tonic seizures that most of the children have. The same for mutations, for example, in this site. These children, they develop normal, or they have some delay in the beginning, and then when they're a few months old, they start to develop spasms and neurodevelopmental problems. The variants at that position, 144. These are all children that do not have neonatal epilepsy. Some of them develop epilepsy later on, and they often have a lot of epileptic activity on the EEG during sleep. Many of these children have very prominent autistic features. And my colleague had tested 2 different variants. The one where the R is changed to a Q was much more severe than the ones where an R, that's the amino acids code, is changed to a W. I talked about retigabine and sodium channel blockers for the variants or patients or children that have variants that lead to a loss of function. Unfortunately, for the children that have these gain-of-function variants, where there's too much potassium channel function, there's very little information. Which seizure medication works better, we do not really know yet. We do know that there are some other drugs that are used for other disorders. For example, amitriptyline, that's a drug that's used for depression or for headaches, enuresis, so this is peeing in bed. Even in children, it's used sometimes. That it also blocks the channel a little bit, KCNQ2. The same for donozepam. That's a medication that's used for Alzheimer's disease. Guanfacine, that's a medication that's used for ADHD. We know that apart from the actions that it has for these other disorders, it also acts on the KCNQ2 channel and it blocks the function. So you would think that maybe it would work for children with these gain-of-function mutations, but we have still very little data on that. I know of a few children that tried amitriptyline. I know of a colleague in Israel that has tried donozepam. Some children seem to do better, others don't seem to respond. Does it depend on the variant? Does it depend on the dose? We do not know yet. But this is something that some children, together with their treating physician, are trying. So this is a little bit a summary. And we know there's a whole spectrum of disorders related to KCNQ2. And it's totally related to how good the channel function. If it functions a little bit less, then you have this benign neonatal familial epilepsy. If it almost doesn't function at all, at the elder extreme, you have these very severe disorder with neonatal seizures and very severe neurodevelopmental problems. And on the other hand, you have the gain-of-function variants when the channel acts too much. And then you have children that generally don't have neonatal epilepsy, but they can develop epilepsy later, but all of them also have neurodevelopmental problems. So if you have a variant in your child and you don't know what it does, because as I told you, it might be important to choose a therapy. Can they go in the trial, for example? The trial, the EPIC trial that I will talk about later, retigabine, it opens the channel. So it's for the children where the channel doesn't work that much. So you need to know what the variant in your kid does. If you don't know, maybe first ask your doctor. Do you know if other kids with this disorder have been described? There is this variant registry, ricky.org. Some of the variants that have been reported and tested you can find there, but not all of them. Or you can contact people that are involved in KCNQ2 research, and feel free to contact me. I have my email address at the end. I try to respond as much as possible. Sometimes I also don't have the answer, but at least I can see, do we know other children? Has the variant been tested before or not? Just as a summary, I try to explain you already something that we have learned over the years, but there's very many remaining questions. There's a lot that we still don't know. For example, I said the variant, it has a big role, but even children with the same variant, they're not totally different. Some have a lot of seizures. In some, the seizures stop after a few weeks. Some can walk and run. Even others, they're wheelchair bound. There's differences, and we think that part might be of the medication they take. For example, if the seizures are controlled very quickly, they might do better. But it might also be other genetic factors. We're all different. We all have a genetic or differences in our DNA. And maybe if you have maybe a variant, I don't know, in a sodium channel or something that makes this work a little bit less, you're better. I don't know. So this is something that we try to look at in our group, for example. And I think some of you already contacted us. So what we do is we collect DNA of the children to look at the DNA, and we look information about the clinics. How well do they do? Do they very good? Do they have a lot of seizures or not? So we need also some contact with the treating physician to give us this information. And then we really want to see, can we find these genetic factors or drugs effects that influence the outcome? We still had talked a lot about epilepsy, and I said it's clear if the brake doesn't work, you get epilepsy, but why, if KCNQ2 doesn't work well, do you get neurodevelopmental problems? We actually do not totally understand yet. It's probably because the nerves don't communicate very well, but do they, I don't know, is it specific cell type? We don't totally understand yet. For the gain of function variants, where the channel acts too much, we also don't totally understand yet why do these children also have epilepsy. What is the best treatment? And a very important question, when do we, for neurodevelopment and not for epilepsy, how much time do we have to cure the neurodevelopmental problems? Imagine that we find a gene therapy in the future, that we can correct the gene. If I give this to an adult kid with KCNQ2 encephalopathy, will he still get better, or is it too late? I think we all agree that the sooner you treat, probably the better. But I still believe, but we don't have evidence, that even later in life, you can still make things better when you find the right treatment. So I think there's all these different types of research going on all over the work. There's still people that look in the dish, but there's still variant that we have to test. We have these iPS cell-derived neurons, for example, in our lab, but also other labs, where we grow from blood or skin cells from a patient, we grow neurons. There's mouse models, as I said, and obviously getting information from all of you and parents, and it really helps to understand this disorder. So at the end, I think we've done already a lot together, and I greatly thank also the German Patient Foundation for supporting all the research. And there's the American parents. It's incredible what they can do and how they can stimulate. Also very important to have researchers and professors and work together, because this is really important. There's so much to do, so we don't have to compete. We have to work together. Also industry, there's a lot of companies that are working or trying to develop these potassium channel openers, maybe blockers. So we also have to work together. But I think, so as I end the conclusion, so both these very severe loss of function variants as gain of function variants in KCNQ2 can lead to neurodevelopmental problems. And whether or not your child had really typical neonatal seizures, it mostly can differentiate between the two types of variants. But I think we all can learn from each other. If you know of other kids with the same variant, contact people in the field to discuss whether we know what the variant exactly does or not. So this is my last slide, and I put my email address there. Of course, we can just talk today for the people that are here, but other people that want to contact us. There's also the email of Charissa, who's also here, a student, and also medical doctor in my team who works with me to try to still resolve a lot of questions that remain. Thank you. Happy to take any questions, obviously, right? Yes. You can even ask them in German. I probably can understand them, and if not, maybe somebody can translate. Or come up to me afterwards. That's perfectly fine. I'll answer it in English, and then maybe somebody, Sebastian, or yeah. So it really, what really helps is knowing which mutation the child has. This is one. Yeah. And what also helps is knowing how his disease history was. Did he or she had these very early seizures with the stiffening and the clonic jerking? Both the combination of these two things and information from other kids that have the same variant, it can really help. And I can check indeed for this variant if we know it Yeah. Yeah. I think so. Indeed, this helps. What I can do is look for this variant in our registry, whether I have information about this variant from other kids or whether it has been tested. So I cannot say it from by head, but I will check it after the in between. That's okay. There's not that many families. That's okay. I can do it in between.
Ich kann das nachher nachschauen, sie hat eine Datenbank und dann kann sie gucken und kann das vergleichen. Du kannst dann nachher, wenn sie fertig ist, mit euren Mutationen hingehen und sie kann jetzt in der Datenbank nachschauen, was da möglich ist, was sie sagen.
Yeah. I have to say, we can say it for many variants. Sometimes there's still doubt. For some variants, we're not sure yet, or if the clinical features are not so very clear. But for many of them, we really can say for sure whether it's the one or the other.
Wie lange ist der Kaliumkanal eigentlich wichtig im Körper? Ich habe gehört, mit dem zweiten oder dritten Lebensjahr ist der gar nicht mehr notwendig. Ist das richtig?
That's a very good question. So, yeah. Yeah. It's indeed so. Now, first of all, KCNQ2 is, because I said the DNA is the same in every cell, but all the proteins can be present in every cell, but our body can arrange that some proteins, like KCNQ2, are present in a cell in a certain time when it's important. Yeah.
KCNQ2 ist halt mit diesen Proteinen, it's a protein for the KCNQ2 channel that
Yeah.
it's working for the system.
Yes. So maybe I should. I'll start differently. We know that KCNQ2 is present in the nerve cell during the whole life, from fetal life, so before birth, until when you die. Yeah, it's there. So, because your body can arrange things, they can make sure that a certain protein is not present. If it's not necessary anymore, it's not present anymore. So the fact that it's there, it tells us that it still does something. If not, it would not be there always.
Dieser Genteil defekt ist, dann bleibt es auch defekt. Und es gibt aber Teile, die können es übernehmen. Some parts can take over.
Yes, and what I wanted to say is the fact that it's there the whole life, it already tells us that it does something. It should,
Es macht irgendwas,
yeah.
nicht das, was es vielleicht soll. Es hat definitiv eine Funktion.
Yeah. But we know that it's most important in the beginning of life. The first months, years of life, it's most important. And why is that? I told you that KCNQ2 is like a break. And very early in life, in fetal life, and in the first months, it's the only break in our one of the only breaks in our brain that's there. Yeah.
Das ist halt ganz wichtig in der ganzen Entwicklung vom embryonalen Stadium bis hin zum Leben. Und gerade in den ersten Wochen, Monaten und Jahren ist gerade das KCNQ2-Gen wichtig für diese Entwicklung, dass es irgendwas unterbindet. It's therefore
To inhibit, yeah, and it's the only protein in our brain that acts as an inhibitor. Later in life, there's other inhibitor, and there's a GABA receptor that only becomes an inhibitor later in life. So then you have two things, so it becomes less important. Yeah, and that's probably why many of the children have a lot of seizures in the beginning, the first weeks, months, years of life, and then in general, epilepsy gets better because you have these other inhibitors that. Yeah.
But that means also that even if there will be a cure, you cannot change the beginning of life. So
yeah, that's a very good question. And so it's there. It does something from already in the womb. So this part you will never be able to change, or you have to make the diagnosis before the child is born. But obviously there's already also other disorders that we know, even if it is a protein that is very important early in life, you can still fix something later in life. Maybe not everything, but something. We know that nerve cells, they connect and make new connections a lot when the brain is formed in the womb. But even until adolescence, when you're 18 years, the brain still develops. And this is a process that we still can influence. I agree, probably the earlier you treat, probably the better. I don't think there's any doubt. We don't know yet how much can we still win when we treat later. I think everyone hopes and is convinced that at least we can still improve something. Just because it still has a function, it's still there to do something. If we can treat that, then we hope that at least we can ameliorate things considerably. No.
It seems to me that all medications are focused on the epilepsy.
Yeah.
Are there any medications for the hypertonic?
Yeah. That's indeed, that's the real need. All medications that we have now, it focus on seizures. And we hope and we know that if you treat seizures, the children will be more awake and they will develop a bit better, but they don't solve all the problems. The only first treatment that we hope has also an impact on treatment is retigabine, ezogabine, that I will talk about next. We hope it because it directly acts on the channel. So it corrects a little bit the problem that's in the channel. We will have to see whether it also then influences the neurodevelopmental problems. But that's the hope. Yeah.
But it's not oxcarbazepine the one who is the key for the channel opening system and does go not for the seizures, then for the channel?
So oxcarbazepine is one of these sodium channel blockers. So they block the channel that's next to it, that's too active because KCNQ2 is not that active. So it mostly suppresses seizures. So it mostly acts for seizures. But it's true that although this is not really proven yet, but we think that if we treat the seizures very earlier, we have the idea that children do somewhat better. But I just say this because I have this feeling when I talk to parents, I see the parents, we don't have proof yet. We want to do that, look at it also in our group. But it's also logic, a little bit logic, right? If they seize every day or every hour, you don't have time to develop. If you correct that, your brain cells can develop better. Right? But it's an indirect result. Yeah.
And what is the function of phenethylamine? Is that
It's also a sodium channel blocker. Yeah, you have different, they act on different positions on the channel, but they're all sodium. So phenytoin, carbamazepine, oxcarbazepine, lamotrigine, lacosamide, these are all sodium channel blockers. Yeah.
So that means that it works good. We don't have to think about changing the medication.
No, and especially if the seizures are controlled, then there's no need to add another seizure drug or whatsoever. Another question is, does he or she has to take this medication for the whole life? There's definitely, as we heard before, there's children that when they're seizure-free for a few years, they stop the anti-seizure medication because we know they only act through stopping seizures. And in some children, seizures go away. As I said, probably we think because these other inhibitory forces, GABA, they act together then. Yeah. So epilepsy becomes less important later in life. I have to add, some children stop and do still have some seizures later in life and they have to restart medication. But most often this epilepsy is not as severe as in the beginning. And some children remain without seizures. Yeah. Yeah. Mm-hmm.
And our variant is not in there. Does it mean it's a new one?
No, not all variants, because this is a public database, and not all variants are on there yet.
Okay.
some are already published, and so I can check whether we do know some because of emails from parents or whatever. Or, yeah, and I think you already did that, right? Yes. Yeah. Yeah. Yeah.
But
Yeah.
the thing we have still for the kids, if we have a mutation in the KCNQ2 gene, and we have it like 10 times, 20 times, same mutation, but all the kids are totally different.
Yeah, that's it. That's the thing, and in general, they're most of the time either on the very good end or somewhere in the more severe. There's some problems, but there is a difference, and that's exactly. We think that it's maybe treatment, as you say. If you treat the seizures very early and very good, maybe they do better, and then maybe our other genetic variants. And we're all different. Maybe you have a variant in, as I said, in a sodium channel that's not disease-causing, but it helps to work a bit better or less. And that's something that we want to do with the study, that we collect DNA for.
Why is it not possible that we have like kids with a mutation and we have a child that's really good? No seizures, doing progress in movement, and and we have parents, they have the same mutation, and it's really, really bad. They have seizures every day. Why don't we check other genes next to this and next to that, and say we have to force this, and for this child especially, we say we have to check much more genes to help this child.
That's exactly what we're trying to do. So if you know these kind of, especially all children are important, because as you say, everybody's different. And so what we want to do is to check all the other genes and see how they differ. But the most interesting, it's a strange word, are exactly those that have the same variant and do so much better or so much worse than all the others, because they have to have something that's different in another gene. So if there's parents that notice this, they're very happy, welcome to contact us, and we will check the DNA.
Is there any plan? You say, okay, if there's a worse child, then we know we have to check this gene, and this gene, or is it like you have to search allele in...
Yeah.
Do you know
For
related
we know related genes. What we're gonna, I'll tell you what we want to do. There's maybe not best strategy, because for now we can have a lot of hypotheses about related genes, for example. But what we want to do is we look at all the genes, we sequence them all. You can do that, it's whole exome sequencing. And then we focus first on the related genes. Do we see differences there? And then on other genes that are in the brain, do we see differences there? But we started immediately to look at all genes because we don't know yet where to look. The changes or the differences might be subtle. So that's why this type of study, it only works when you have either a lot of children that contribute, because we are all different. We will find differences, but are they relevant? Or when we have children that are really different, like you said, one that does very well and the other's bad, or everybody does good and then this one does bad. Those, maybe they have something very important that doesn't go well or better, or yeah.
Gene therapy. I wrote a little about this ASO, antisense oligonucleotide treatment. What do you think today? What would be the best solution for the future to maybe correct effect?
Yeah, I agree with you. I think the antisense oligonucleotides, they're a feasible and realistic strategy to pursue. Whether it will help, we will have to see, obviously, right? But for example, there is now a clinical trial with an ASO, an antisense oligonucleotide. Maybe I have to explain what it is. An antisense oligonucleotide is a small molecule that looks a little bit like DNA. It's actually RNA, and it can bind to the RNA of KCNQ2 in this case, to reduce its expression or to increase its expression. Depends where it binds. So it's kind of genetic therapy. It doesn't work on the protein, it works higher, it works on the DNA level. It's a very new field. There's some antisense oligonucleotides that are already used for other genetic disorders, mostly in the neurology for neuromuscular disorders. But there's now a clinical trial in Dravet syndrome. You might have heard of it. So this is also very severe epilepsy with neurodevelopmental problems, with mutations in the gene SCN1A. So there are trials with azoles already. So it's realistic, it's feasible. We will have to see whether it works, but they're being developed. Also for KCNQ2, there's many people working on it.
So I thought I would say it's like a patch, what you put on the incorrect
Yeah.
gene. And the question is, will the correct gene take the work for the whole system?
Yeah.
So will it work? Or what do you think?
In theory, there's children with BFNE, so just the neonatal seizures, and they miss one copy of the gene. They only have one copy. That's the mutation, and they function well. So if it would be possible that we can take away the bad copy, so make the azole so specific it only blocks this copy, then it should work. Obviously, then we come to the question: how quickly do you have to treat it? But in theory, it
Means for every child, right? For every mutation.
for every variation.
For every
Or you look at variations that are in the neighborhoods that are.
But the best would be for every child or for every variant.
Yeah, but that becomes very expensive. I mean, everything's possible. Or you look at variants that are more common, because we all have variants, right? And that are in the neighborhood. That's another strategy that we're trying to pursue. But yeah, the difficulty is to make an azole that's so specific that it only recognizes that copy and not the other copy, but also not something else in the genome. That's the difficulty, yeah.
What about stem cells therapy? Does it make sense?
Stem cell therapy, there's research mostly because this is something you can plant in the brain. It's not that easy as that, but then they take over. But obviously, KCNQ2 is in every cell of our brain. Then it should go everywhere, let's say, and it also in the spine, because there's also KCNQ2 there. It should make the right connections. So I'm not really convinced this is a strategy for a genetic epilepsy that's everywhere. It's mostly used, for example, for people that have a brain lesion, like after a trauma, and there's a part of the brain that doesn't work well, and then they can implant there better cells, stem cells. Or that have epilepsy from a very focal part in the brain, so you only have to inject or correct something locally.
Also can have the same problem when parents get
Yeah, that's a very good question. Most of these kids have de novo variants, so that means not a mother and not a father. So the chance that the next child has the same problem is very small. Yeah, then he will not have it later either. Yeah. Well, I was going to say the chance is very small. It's not totally zero. And why is that? It's still theoretically possible that the mutation is, for example, only present in a sperm cell or oocytes of the mother, or in a few cells of the body of mother or father, and you don't detect it in blood. That's a theoretical possibility, and that's why sometimes you see two children in families with KCNQ2 or Dravet syndrome, but it's like 0.1%, so it's very small. If you really want to exclude it, if you say, I want to be 100% sure, there exists something like prenatal or pre-implantation diagnostics, then you're 100% sure. But this is something that you can discuss with your genetics department. So the chance is very small, but it's not totally zero.
But
Yeah.
no family with de novo, which they have more than one
Yeah,
kid,
yeah.
they have problems.
Yeah, yeah,
Kids with de novo.
yeah.
I think 3 kids. There's one
What, is
family.
there about 3? I remember
Yeah,
2 that there were.
there were twins. And
Oh,
one-
twins. Yes,
they
yes,
had twins
yes.
and one
Yeah.
baby, and all 3
Yeah.
were diagnosed with KCNQ2 with problems.
So it's not only for casing. There's just the mutation has to start somewhere, right? And most often it's when the embryo is made, but it can also, the mutation can occur in a sperm cell or in a few cells in the body of a person that you can't detect in blood, and then of course you can transmit it to the next generation. Yeah. So the chance is very small, but it exists for all genetic disorders that are de novo. Yeah.